Hello, my name is Jordan Hinds, and under the mentorship of Dr. Jones Sartori, my project is using direct-fed microbials, Saccharomyces cerevisiae, to increase cattle feed utilization when undergoing distinct feeding management, an in vitro approach. The use of yeast in finishing and growing diets were the focus of this project, as it may provide optimum outcome when implemented. A 2x2 factorial arrangement of treatments were used, with the two main factors being A, the presence or absence of yeast, and B, two digestive tract conditions, either a grower or finisher diet. The project focused on the interaction of the yeast and ruminal environment pertaining to in vitro true digestibility of dry matter, organic matter, and fiber components. The main goal of this project is for the cattle to better utilize the nutrients present in the diet that they are consuming. By improving the efficiency of the digestive tract, this will allow the animal to excel in other areas that producers deem desirable, such as average daily gain. Dietary expenses represent approximately 70% of production costs, and implementing an ingredient that improves the efficiency of the digestive tract of cattle will benefit producers in the long run, as the animal will operate more efficiently and may provide additional relief or greater profitability. The materials for this project consisted of 4 ruminally cannulated beef steers that served as our rumen fluid donors, 12 beef cattle diet specimens that were collected from feedlots located in the Texas Panhandle, and 3 individual ingredients, which were steam flake corn, wet corn gluten feed, and Old World blue stem hay. In this video is one of our cannulated steers during the collection process. His cannula is open and you are able to view the rumen and fluid inside of it. First, our cannulated beef steers were adapted to a specific diet, grower or finisher, for 7 days prior to the collection of ruminal fluid. After ruminal content was collected, McDougall's buffer was mixed with ruminal content at a 4:1 ratio. F57 Ancon bags containing pre-dehydrated diet specimens and ingredients were incubated for 48 hours at 39 degrees Celsius. The ruminal incubation batches were replicated 4 times, in which the initial 2 batches were performed, followed by another 2 after animals switched diets and were adapted again. It was a crossover design, as there were 2 animals on a 50-50 diet and 2 on a 92-8 diet, which eliminated any animal implication or confounding. Listed are the ingredients of our grower diet, which contains 50% concentrate, the percentage of dry matter abbreviated as DM of each ingredient, along with the percent inclusion on a dry matter basis. Next is our finisher diet, which contains 92% concentrate. The ingredients are listed along with the percentage of dry matter and the inclusion of each ingredient on a dry matter basis. In this slide is our grinder, which was used to grind the 12 diets and individual ingredients to a particle size of 1 millimeter. We then placed 0.5 gram of the respective sample in an F57 Ancon bag. Pictured are the F57 Ancon bags that were used for this project. In the first picture is the process of filling them, the second photo is how they are sealed, and the final photo is the final product before they are placed in the Daisy incubator. All bags are labeled and weighed prior to being placed in the incubation chambers. Pictured is our Daisy incubator that was used. The in vitro yeast dose was adjusted to represent a 70-liter ruminal content volume. The bags are incubated for 48 hours at 39 degrees Celsius to ensure continuous control of temperature and movement. As mentioned earlier in the introduction, the project focused on 5 different areas of in vitro true digestibility. The first one is dry matter, and the results are shown in the graph. The digestibility of dry matter was greatest in the 50-50 ruminal content that contained yeast and was 83.5%. The next area of focus was organic matter. The greatest result was yielded when yeast was added to the 50-50 ruminal environment, showing 83.03%. For neutral detergent fiber, the ruminal environment of 50-50 with the addition of yeast yielded the greatest result of 47.56%. For acid detergent fiber, otherwise known as ADF, the ruminal content did not alter the yeast's effectiveness, as both were improved with the addition of yeast in both the 50-50 and 92-8 ruminal environment. In our last graph, hemicellulose was greatly improved in the 50-50 ruminal environment with the addition of yeast, showing a result of 49.13%. Conclusion: the inclusion of yeast seems to improve ADF digestibility regardless of nutritional management, either a grower or finisher diet. More intense improvement in ruminal degradation was observed when yeast was included on a grower diet ruminal inoculum, which was the 50-50 diet. I would like to thank Dr. Jones-Sarturi and the members of the Ruminant Nutrition and Metabolism Lab for the time and effort they put towards assisting me with this project. I would also like to thank the Center for Transformative Undergraduate Experiences and the Department of Animal and Food Sciences at Texas Tech for their generous contributions.